In this lecture, we're going to look at three families of viruses, all DNA viruses: the polyomaviridae, the papillomaviridae, and the adenoviridae. Now, if you think back to the previous lecture about the parvoviruses, remember that they were very tiny viruses and they were limited to actively dividing cells. Bigger DNA viruses encode proteins that induce resting cells to begin dividing. It's been shown that RB protein is responsible for a major G1 checkpoint, blocking S phase entry and cell growth. P53 is a transcription factor that is induced by many stress signals such as DNA damage, oncogene activation, and nutrient deprivation. Cell cycle arrest and apoptosis are the most prominent outcomes of P53 activation. So the goal of these three viruses is going to be how to get around those checkpoints, the RB protein and the P53. And what we find is that the early proteins that are produced upon infection of a host cell play a large role in interacting with these two proteins and allowing the cell to enter into cell division. So the first virus family we want to consider is the polyomaviridae. In terms of characteristics, the virions are non-enveloped with an icosahedral capsid. Each virion contains a single copy of circular double-stranded DNA. The mature viral genome is organized as a microchromosome packed with histone proteins. So if you look at that picture at the top right, those round structures are actually cellular histones that are associated with the mini chromosome of the virus. Transcription from one side of the viral origin of DNA replication, or the ORI, results in messenger RNAs that encode the early proteins. These non-structural proteins are referred to as tumor antigens, as they interfere with cell cycle regulation and, in some cases, induce cellular transformation or tumor formation. So remember, DNA viruses, when we consider the replication cycles, they can be divided into early and late stages. An early stage, of course, is absorption, penetration, uncoating. And then you have the transcription of the early genes producing the early proteins. And in this case, the early proteins are called T antigens because they stimulate the production of enzymes that are required for cellular DNA replication. After DNA replication commences, then you shift to the late stage, and that's when you have replication of the viral DNA and expression of the late genes, which are going to be structural proteins, and then assembly and release. The principal function of the early proteins is to enable viral DNA replication. Most cells in an organism are not dividing, but are differentiated and in a resting state, the G sub 0. Under these conditions, the enzymes and precursors needed to support cellular or viral DNA synthesis are not normally present in the cell nucleus. The early proteins of DNA viruses induce entry of the cell into the DNA synthesis, or S phase, of the cell cycle. Now, we can look at this. One of the best studied viruses here, the BK polyomavirus, is a naked, circular, and double-stranded DNA virus. The genome is organized into three regions: there's a non-coding region, the NCCR, which contains the origin of replication, or the ORI. The transcription promoters and enhancer regions are also found here. The early structural region encodes two non-structural proteins, the large T antigen and the small T antigen, that are responsible for viral replication and gene transcription. The late structural region encodes three capsid proteins, VP1, VP2, and VP3. So again, you have the virus being brought in through receptor-mediated endocytosis. The DNA is released into the nucleus of the host cell. Early messenger RNA is transcribed by a cellular RNA polymerase 2, and the T antigens, the early proteins, are translated. As concentration of large T antigen increases, the transcription of early genes is repressed, and you get a switch to genome replication. In terms of genome replication, remember the switch from transcription of messenger RNA and translation of protein to genome replication is initiated by the binding of large T antigen to the origin of the genome. So as the concentration of large T antigen increases, this switch is going to occur. The protein-protein interactions between T antigen and DNA polymerase directly stimulate the replication of the virus genome. Inactivation of tumor suppressor proteins bound to T antigen causes G1-arrested cells to enter S phase, promoting DNA replication. The DNA synthesis is bidirectional. When the replication forks meet, the daughter genomes separate. Okay, so now we move on to late events in infection. The large T antigen, in addition to initiating DNA replication, regulates the transcription of late messenger RNAs. And remember, the late proteins are going to be structural proteins. After you have the DNA inserted into the assembled capsids, the virions are transported to the cell surface in vesicles. Now, there are two different ways that the infection can go, depending on whether or not it's a permissive cell or a non-permissive cell. So let's look at that. In a permissive cell, the large T antigen interacts with the cellular regulatory proteins RB and P53. And remember, the RB protein controls the cell cycle and S-phase gene expression, and P53 blocks the cell cycle and induces apoptosis in response to viral infection. So the large T antigen interacts with these two proteins to prevent that and allow the cell to move into division, into the S phase. So binding of the large T to RB stimulates the cell cycle. Binding to P53 allows the cell to enter the S phase while avoiding the P53-mediated apoptosis. So you get productive infection. The infection is lytic. You get expression of late genes and assembly of progeny virions, and ultimately this leads to death of the cell. When the virus infects non-permissive cells, you get a non-productive infection. And in this case, you have the large T antigen binding with RB and P53. This allows for the cell to move into the S phase so that you get division, and it blocks P53 so that you don't get apoptosis of these dividing cells, and you get division of the cell because P53 and RB have been regulated. They've been downregulated. And in this case, what happens is, because it's a non-permissive cell, the virus can express its early genes, so the large T antigen is expressed, so that can allow the cell to move into the S phase, but it can neither replicate its DNA nor express late proteins. And so no progeny virus is produced and the cell is not killed. In this case, you can have transformation of the cell. If we look at these biological properties, then we can look at what happens when you have a productive infection in a permissive cell and then a non-productive infection. So, of course, with the productive infection, you get DNA replication, you get transcription and translation of the late proteins. That allows for the assembly of progeny virions, and ultimately you get death of the cell. With a non-productive infection, that's what occurs with non-permissive cells. Remember, DNA replication cannot occur, but the transcription of messenger RNA and translation of early proteins, specifically the large T antigen, is happening. And so that can interact with RB and P53. The viral T antigens and their interaction with P53 and RB allow the cells to acquire the properties of transformed cells. Usually this transformation is a transient thing, and it becomes abortively transformed. The genome can't replicate, so eventually it's lost from the cell, and the cell returns to normal. But occasionally, the viral genome can integrate into the host genome. This occurs at low frequency, but it is a possibility, and that can lead to a permanent transformed phenotype. Okay, so now let's look at some of the diseases that are caused by polyomaviruses. The pathogen avian polyomavirus is considered one of the most significant threats to caged birds around the world. This highly infectious disease affects most, if not all, parrot species. Polyoma seems to be most problematic amongst young birds between the ages of 14 to 56 days old. These young birds often die, while adult birds can develop a certain level of immunity. Polyoma is believed to have an incubation period of approximately 2 weeks or less. The disease can be spread from one bird to another through feather dust, feces, aerosols, and parental feeding of the chicks. APV can also be transmitted by direct contact with infected environments, such as incubators, nest boxes, and so on. Birds that are infected but do not have obvious signs of infection are often responsible for spreading the virus to an aviary or a bird store. So adult birds can be carriers of APV. They don't necessarily show symptoms, but they're able to pass the disease on to others. So it's important to isolate all birds that are shedding the disease. You have to disinfect all contaminated surfaces with an oxidizer such as chlorine bleach Just like with parvovirus, polyomaviruses are resistant to many disinfectants. For instance, alcohol will not work against avian polyomavirus. A vaccine is available, but this option can cost as much as $50 per bird. And also, booster shots are required each year. And how effective the vaccine is in younger birds is in question. So the best thing is to quarantine all new birds, and there are tests that can be done, including a PCR test, to determine if a bird is infected. There are human polyomaviruses as well. We'll talk about BK and JC viruses. Each of them got their names from the initials of the patient from whom the virus was first isolated. For both viruses, the infections are acquired early in childhood, and adults in the U.S. population are largely seropositive, so these viruses are ubiquitous. Now, the majority of infections lead to latency in the kidney, and they're subclinical. The big problem is when there's reactivation, and that can occur under circumstances of immunosuppression. So, in the case of BK virus, the virus may become activated in various situations of impaired immunocompetence, such as solid organ transplantation, bone marrow transplantation, AIDS, pregnancy, multiple sclerosis, chemotherapy, and so on. And nowadays, with the use of potent immunosuppressive agents and enhanced viral surveillance protocols, the BK virus has arisen as an important cause of morbidity in renal transplant recipients. So, in this case, with BK virus, it's latent in the kidneys, but when it's reactivated, its major target would be the kidneys and the urinary tract. So, with these individuals who go in and get an organ transplant, particularly a kidney transplant, there's sort of a conundrum for the medical personnel who are managing the patient, because current therapies aim to restore the impaired immune response. So they actually use immunosuppressive agents to prevent rejection of the transplanted organ. However, if they reduce the amount of immunosuppressive drugs that are given to the patient, that can reduce the possibility of activating BK virus. So it's kind of a double-edged sword, since if you reduce the amount of immunosuppressive drugs, then it increases the chance of rejection of the organ. The AIDS epidemic of the 1980s led to a dramatic increase in new cases of PML, which helped fuel research towards the mechanism, risk factors, and possible therapeutic interventions for this devastating disease. So, as we were talking about before, just like with BK, JC virus persists within the kidneys and lymphoid organs of healthy individuals. Progressive multifocal leukoencephalopathy, or PML, is a slowly progressive demyelinating disease of the central nervous system, and it's a fatal illness that's caused by reactivation of JC virus in immunosuppressed hosts due to the loss of protective cellular immunosurveillance. People with HIV/AIDS are most at risk for PML. Those with Hodgkin's disease, leukemia, or lymphoma are also at risk, as are people who take certain immune-suppressing drugs because of multiple sclerosis, rheumatoid arthritis, or organ transplants. So here we've got the flow chart looking at both JC and BK viruses. These viruses are believed to enter through the respiratory tract through inhalation. You get initial multiplication in the respiratory tract, the primary viremia, and the virus disseminates, particularly to the kidneys. You get a secondary viremia, and then if the individual is immunocompetent, it can remain latent indefinitely for the life of the host in the kidney. But if the patient becomes immunodeficient, you get reactivation of the virus. Now, if it's JC virus, the reactivation leads to targeting of the central nervous system and the development of PML. If the virus is BK, when it's reactivated, it multiplies in the urinary tract, and you get various types of damage to the kidneys and urinary tract. And again, this BK is especially a problem for individuals who have received kidney transplants, because they're going to be taking immunosuppressive drugs, and the transplanted kidney is a perfect target for BK virus. So now we want to move on to the next virus family, the papillomaviridae. These are non-enveloped viruses. They're small, icosahedral. They have a circular double-stranded DNA genome. And just like with polyomavirus, this double-stranded DNA is associated with cellular histones in a chromatin-like complex. In fact, papillomaviridae and polyomaviridae were at one time in the same family, so there are a lot of similarities between these two virus families. There are more than 150 different types of HPV, each with its own favorite skin surface to invade. Some cause the small, painless, rough-surfaced warts found on the fingers and face. Others cause the larger, more painful and flatter plantar warts that grow on the soles of the feet. More than 40 different types of HPV can infect the skin covering the sex organs, cervix, and opening of the anus. Genital HPV infections are very common. Up to 80% of sexually active adults will get an HPV infection of the genital area at some point in their lives. In most cases, these infections do not cause symptoms, but they can cause genital warts. In a small number of women, certain HPV strains cause changes to the cervix that can become cancerous if not treated. HPV is also linked to other cancers, including cancers of the penis, vagina, anus, vulva, and also to the mouth and throat. Human papillomaviruses are usually spread by direct skin contact, such as shaking the hand of someone who has a wart on their finger, or having sexual intercourse with someone who has a genital HPV infection. Genital HPV infections can be spread by people with no symptoms, but the risk of infection is particularly high if you have intercourse with someone who has genital warts. So, my advice there, keep the lights on, gang. Less often, the viruses are carried on surfaces touched by someone who has warts, especially inside shoes that have been worn with someone with plantar warts. So there, wear socks when you rent shoes at the bowling alley. Once a person has been infected with an HPV infection, symptoms usually take 3 to 4 weeks to develop. However, in some cases, warts have developed as long as 2 years after contact with an infected person or contaminated surface. In terms of viral replication, just as with the other viruses we've talked about, you can divide the cycle into early and late stages. In this case, the replication of the virus is linked to the differentiation state of the epithelial cell. These viruses infect the stem cells, the skin and mucous membrane, so epithelial tissue. They undergo their full replication cycle only in terminally differentiating epithelial cells. So let's take a look at this. Following viral entry and uncoating, HPV genomic DNA is transported into the nucleus and maintained at a low copy number in the basal cells, anywhere from 50 to 100 copies per cell in the basal layer. And if you take a look at this illustration, you can see the structure of the epithelial tissue here. So remember, the basal layer is the only layer that's actively dividing, and then it pushes up these other cells that become differentiated. Genome maintenance as an episomal, in an episomal status is essential for the establishment of the early phase of the viral life cycle. After leaving the basal membrane, the infected cells initiate the differentiation program. Because HPV does not encode DNA polymerase activity for viral genome replication, the host DNA replication machinery is required. However, the DNA replication activity is suppressed in cells that have differentiated, so HPV needs to reactivate cell division among the differentiation-initiated cells. E6 and E7 inactivate P53 and RB protein respectively, which enables the cells to maintain their DNA replication potential. In the upper layers of the stratified epithelium, the expression of viral genes that are required for viral genome replication is markedly accelerated, inducing viral genome amplification to thousands of copies per cell. Following the genome amplification in the terminally differentiated cells, the synthesis of capsid proteins is triggered. The capsid proteins assemble into virions that encapsulate the viral genomic DNA. The progeny virions are released externally with the keratinocytes that are sloughed off. With this slide, we're looking at in situ hybridization, and we can see the expression of different proteins in these sections of tissue. So A, top left, that's control, that's normal tissue, that's what it would look like. In C, you have staining using a probe to one of the late proteins, L1. You're probing to its ORF, so that you can see that that's where you get gene expression of these late proteins in the differentiated cells. So you can see that it's up near the surface of the skin. If you look at D, the probe is to one of the early genes, E5 in this case, and you can see that that's primarily expressed in the basal cells. So the replication is tied in to the stratified nature of these cells. The early proteins, E1 and E2, direct initiation of DNA replication. E1 is a DNA helicase that binds to the origin of replication and unwinds the viral DNA. It's a homologue of the polyomavirus large T antigen. E2 acts as a transcriptional regulator. Virions are taken up by endocytosis, and viral DNA enters the nucleus and is maintained as a free circular mini-chromosome in the basal cells. Early genes are transcribed, permitting an initial replication phase that results in 50 to 100 copies of DNA per cell. Genomes then replicate on average about once per cell cycle as the basal cells divide and the viral genomes are distributed equally among the daughter cells. This is referred to as plasmid replication. When the basal cells become committed to the differentiation pathway that epithelial cells go through, there's a burst of viral DNA synthesis known as vegetative replication. At the same time, L1 and L2 are expressed, producing the virus capsid proteins. Progeny virus particles are assembled in the nucleus and are released upon cell death and shedding at the surface of the lesions. Although at least 3 viral promoters are used to initiate transcription, viral mRNAs are largely the product of processing of pre-mRNAs by alternative splicing and polyadenylation. The HPV life cycle and viral gene expression are tightly linked to differentiation of the epithelium that the virus infects. There is an orchestrated production of viral mRNAs and proteins. The viral DNA is chromatinized by the recruitment of cellular histones. Remember, we talked about that a few slides back. And these are subject to host cell mediated post-translational epigenetic modification, recognized as an important mechanism of virus transcription regulation. The HPV life cycle is dependent on the terminal differentiation of the target cell within the epithelia. This would be in the keratinocyte. The viral life cycle begins in the undifferentiated basal compartment of the epithelia, where the viral chromatin is maintained in an epigenetically repressed state, stabilized by distal chromatin interactions between the viral enhancer and the early gene region. Migration of the infected cell towards the surface of the epithelium induces cellular differentiation, which disrupts chromatin looping and stimulates epigenetic remodeling of the viral chromatin. These epigenetic changes result in enhanced virus transcription and activation of the virus late promoter, facilitating transcription of the viral capsid proteins. So let's look at pathogenesis and pathology. Most often, infection with a papillomavirus results in a benign epithelial lesion. These are often on the skin, we would consider them warts, and if they're on the genitalia, we use the term condylomas. And it requires infection of the basal cell. Remember that that's where the initial replication of the viral DNA is going to occur as an episome. And so oftentimes the infection of the epithelial tissue is associated with some sort of minor injury, a scratch or something that allows the virus access to the basal epithelial cells. But then once they've infected the cell, then you get DNA replication and ultimately movement and replication of the virus as the cell differentiates and moves to the surface, and the virus will eventually be sloughed off along with the dried, sloughed off cells. The papillomaviruses are resistant to desiccation, and transmission through fomites can occur. So with most HPVs, that's what we have. It's a benign type of lesion. It may be unsightly, but it's benign. But there are certain types of HPVs that can cause progression to malignancy. And that's what we want to talk about next. So with certain types of HPVs, you can have dysplasias and carcinomas resulting. Most of the time, these cancers don't occur until many years after the initial infection. The immune status of the individual plays an important role. There are exogenous co-carcinogens, such as smoking and other health aspects of the host, that play a role in determining whether or not it will become malignant. And also the viral genotype plays a role in the progression. Types 16 and 18 are particularly high risk for cervical cancer. But one thing that is critical in this step is that you have to have integration of the genome of the virus into the host chromosome. And remember, E6 and E7 circumvent normal control of cell proliferation, so they play a role in this manipulation. Papillomaviruses, remember, do not code for their own DNA replication enzymes. Therefore, in order to replicate their DNA, they must use the machinery of the host cell. But we do see viral DNA replication occurring in fully differentiated keratinocytes, which do not normally divide, and therefore you don't have DNA synthesis normally occurring in these cells. It's the role of E7 to induce cellular DNA synthesis. It targets RB protein, which is a major regulator of the cell cycle, and functions through its ability to bind to and repress the activity of a group of cellular transcription factors known as the E2F family. When E2F is freed from RB, it can induce the expression of a number of genes whose products initiate and carry out cellular DNA synthesis. Activation of E2F moves the cell cycle from G1 to S. The major role of E6 is to direct the degradation of P53 just in order to inhibit apoptosis in the infected cell and allowing cell survival until sufficient new progeny virus is made. P53 is normally present at low levels in the cell in a latent, inactive form. When the cell is exposed to stressful conditions, such as virus infection or DNA damage, P53 becomes stabilized by phosphorylations and its level increases. The accumulated P53 either induces cell cycle arrest in G1, allowing sufficient time to repair damaged DNA, or it induces apoptosis, which eliminates abnormal cells. Expression of the HPV E7 oncoprotein alone leads to deregulation of cellular proliferation by inactivating RB and other factors, which can be counteracted by P53-induced apoptosis, therefore eliminating growth deregulated cells. So that's the upper path in this figure. But if you have the concomitant expression of E6 inducing the proteolytic degradation of P53, then the apoptotic response towards E7 is blocked, allowing the outgrowth of cells under the abnormal growth stimulus exerted by E7, and that's the lower path in this figure. Soon after the discovery of the first oncogenic HPV type, data accumulated which indicated that the viral E6 and E7 proteins are the key factors that maintain the malignant phenotype of the HPV-positive cancer cells. Specifically, E6 and E7 are the only viral genes that are always retained and expressed in HPV-infected cells. Human papillomavirus-infected cancers are expected to remain a major health problem worldwide for decades. The growth of HPV-positive cancer cells depends on the sustained expression of E6 and E7 oncogenes, which, as we just saw, need to act in concert. But the important thing about knowing about E6 and E7 is that they now constitute attractive therapeutic targets. If you can inhibit E6 and E7, then that inhibition can rapidly induce senescence in HPV-positive cancer cells. The development of cancer from HPV-infected cells is triggered by E6 and E7, as we said. E7 is one of the first genes to activate once the viral genome is incorporated into the host cell. The protein expressed from this gene binds to RB, a protein which can stop cell division by binding to the transcription factor E2F. With RB unable to inhibit E2F, the cells express proteins which encourage DNA replication and cell division. The immortalization of cells is one of the changes necessary for the development of cancer. And then, of course, we have the role of E6 coming in and affecting P53. So there's more than 150 related viruses in the papillomaviridae. Each member of the group is given a number, which is called its HPV type. And if you look at this chart, you can see they can be divided into two groups based on the type of tissue they infect, whether it's a mucosal type tissue, such as the epithelial tissues that line organs and cavities of the body, or cutaneous, the more superficial coverings of the body, the skin, and so on. The cutaneous viruses can cause warts. The mucosal types can cause condylomas, and many of these are low risk, but we have some that fall into the high risk types, particularly types 16 and 18, although those aren't the only high risk types. And with those, there is the potential for cancer. An HPV-associated cancer is a specific cellular type of cancer that is diagnosed in a part of the body where HPV is often found. These parts of the body include the cervix, vagina, vulva, penis, anus, rectum, and the oropharynx, that's the back of the throat, including the base of the tongue and tonsils. These cellular types include carcinomas of the cervix and squamous cell carcinomas of the vagina, vulva, penis, anus, and rectum. Researchers use cancer registry data to estimate the number of HPV-associated cancers in the United States by looking at cancer in parts of the body and cancer cell types that are more likely to be caused by HPV. Cancer registries do not routinely collect data on whether HPV is in the cancer tissue. CDC studies have reported the number of HPV-associated cancer cases per year, and these studies have more information on how HPV-associated numbers were calculated. But you can see from this chart the different types of body tissues that are susceptible to the types of HPV that are high risk for developing carcinomas. And if you look at the bottom, about 91% of all cervical cancers are believed to be caused by HPV, and about 91% of all anal cancers are believed to be caused by HPV, and 72% of oropharyngeal cancers are caused by HPV. There are 3 vaccines approved by the FDA to prevent HPV infection: Gardasil, Gardasil 9, and Cervarix. All 3 of these vaccines prevent infections with HPV type 16 and 18, which are the 2 high-risk HPVs that cause about 70% of cervical cancers and an even higher percentage of some of the other HPV-associated cancers. Gardasil also prevents infection with types 6 and 11, which cause 90% of genital warts. Although 6 and 11 are not really high risk in terms of cancer, they can cause unnecessary concern about the possibility of cancer and lead a person to go in and get various kinds of tests and treatments because they need to find out whether it's one of the high risk groups. So it's good to eliminate 6 and 11. And Gardasil 9 prevents infection with the same 4 HPV types in Gardasil, plus 5 additional high risk HPV types 31, 33, 45, 52, and 58. Now, Cerevix has recently been removed from the U.S. market due to a decrease in product demand. So there's nothing wrong with the Cerevix vaccine. It's just that it's limited in scope. It only protects against infection with HPV 16 and 18, whereas Gardasil and Gardasil 9 protect against a broader array of virus types. So if you were selecting a vaccine, why would you choose the one that only protects against 2 when you can choose one that protects against more viruses? And on the slide, it outlines the reasons why you would want to get an HPV vaccine for a child. It's usually recommended that around the time the child is 10 to 12 years old, that would be the best time to get them the vaccine. You want to get them vaccinated before they become sexually active. So now we want to move on to the adenovirus. The adenovirus has a virus-coded terminal protein covalently linked to the 5' ends. The viral DNA contains inverted terminal repeat sequences of approximately 100 base pairs. This means that the 5' and 3' ends of each strand are complementary and can base pair, allowing single DNA strands to circularize, forming a short panhandle end. Both the terminal protein and the inverted termini repeats are involved in viral DNA replication. So it's a naked virus with icosahedral symmetry, but notably it has those distinct fibers that project from the 5-fold axes of the icosahedron. In terms of an overview of replication, we have, again, an early phase that occurs before DNA replication, so absorption, penetration, the movement of viral DNA to the nucleus, and expression of early genes. Then we move into the DNA step, DNA replication step, and then late is the expression of the late genes and assembly of progeny virions. So again, the first step in the infection cycle is gaining access to the host cell. The 12 spikes of the adenovirus capsid are adhesion receptors which recognize and bond to specific glycoprotein receptors on the target cell membrane. These bind to a glycoprotein on the target cell called CAR, cysteine aspartate protease, or cysteine-dependent aspartate directed protease. This initial adhesion is temporary and insufficient, but integrins on the target cell surface recognize and bind irreversibly to the penton at the base of the spike. The integrins are glycoproteins involved in cell adhesion and cell signaling. In this case, binding of adenovirus to the integrin causes the virus to be taken up by the cell, which is fooled into treating the virus as a normal integrin ligand by coated pit. So it's a type of receptor-mediated endocytosis. The cell surface membrane invaginates, forming a pit, which is coated on the cytoplasmic side by molecules of clathrin. This pit invaginates and pinches off as a vesicle in the cytoplasm, and it contains the virus particle inside. So pretty typical. In this case, it's the penton, the spike, that binds, but it's then your typical endocytosis that brings it in. When the endosome becomes more acidic, this triggers uncoating of the virus. The outer capsid dissembles, revealing the viral DNA protein core. The shed spikes have a toxic function and breach the membrane of the endosome, allowing the viral core to escape into the cytosol of the host cell. The virus then uses the microtubules of the host cell to carry it to the nucleus, where it arrives at and interacts with the nuclear pore complex, or NPC. The NPC is a complex protein machine or gate that controls the entry and exit of materials to and from the nucleus. The virus triggers this gate to open, and its naked DNA enters the nucleus. Once inside, the DNA associates with the host's histone proteins, behaving like host DNA, and then transcription and synthesis of early proteins begins. These proteins inactivate host defenses and synthesize viral DNA. The first viral protein produced, E1A, activates the other adenovirus promoters, resulting in the transcription of the early genes, E1B, E2, E2B, E3, and E4. So as we look at the activities of the early viral genes for the adenoviruses, we're going to see a pattern very, very similar to what we just talked about with the papillomaviruses. E1A is the first gene to be transcribed. The E1A proteins are powerful transcriptional activators of a number of viral and cellular genes. The natural target cells of adenoviruses are terminally differentiated, non-dividing epithelial cells. E1A proteins induce non-dividing cells to enter the S phase of the cell cycle. E1A proteins bind to RB and activates E2F. E2F can then activate expression of S phase genes. Expression of E1A leads indirectly to the activation of P53. And as we've seen before, activation of P53 is a signal to the cell that something is wrong, and activated P53 induces growth arrest and cell death by apoptosis. E1B proteins suppress E1A-induced apoptosis, allowing virus replication to proceed. So E1A and E1B function the same way that E7 and E6 do in the papillomaviruses. Virus-associated RNA are involved in translational control by inhibiting activity of the cellular double-stranded RNA-dependent protein kinase, PKR, one of the pathways by which interferon acts. On the first slide about adenoviruses, I said that there's a protein called TP, or the terminal protein, attached to the 5' end of each strand. Remember, it's a double-stranded linear DNA molecule. I also said that they're inverted terminal repeat sequences, so that the 5' and 3' ends of each DNA strand are complementary and can base pair, allowing single DNA strands to circularize, forming a short panhandle end. Now you're going to see why this was important. So replication occurs. Only one strand will serve as a template. TP will act as a primer. This will result in a duplex molecule, a daughter and parental strand, and a displaced single strand of DNA. So that's what you see down by D. You've got a duplex strand, and the green is the newly synthesized strand, a red is a parental strand, and then the blue is a parental strand that was displaced. Well, what will happen next is that displaced strand will circularize, forming the panhandle, and that allows it to be recognized by the same initiation machinery that operates in the first stage of replication. And you end up with two strands. Each one of them is a duplex of one daughter and one parental strand. Early in the infection, the major late promoter, or MLP, expresses only a small subset of messenger RNAs and is largely suppressed. Consequently, expression of late proteins is low in the early phases of replication. However, this promoter becomes activated with the commencement of the viral genome replication, and it leads to high levels of late protein expression and accumulation of all late transcripts. Expression of late proteins is also regulated by products of the L4 gene, which activates late protein expression after initiation of viral genome replication. E1A plays a role in the regulation of expression for all early transcriptional units, as well as the MLP. In addition, viral non-coding RNAs are expressed early in infection utilizing the cellular RNA polymerase III, while all other viral transcription is performed by the cellular RNA polymerase II. Overall, the virus carries out a highly orchestrated transcriptional program that converts a non-permissive cell into a permissive one in an efficient manner and within hours of infection. Empty capsids are assembled from hexon trimers and pentons, along with the minor capsid proteins and the non-structural proteins. Packaging proteins bind to the packaging domain located close to the end of the genome. The genome is associated with TP at each end. The genome is inserted into the empty capsid through an unknown portal located at the unique vertex. Core proteins are incorporated into the empty capsids during or after genome incorporation. Then scaffolding protein and other non-structural proteins are released. The virus-encoded protease triggers conformational changes in the capsid structure, leading to maturation of the virus particle. Progeny virions are released by host cell lysis to neighboring cells and fluids. This process of lysis is facilitated by a protein called adenovirus death protein, or ADP. ADP is an E3 protein, and it causes membrane rupture. Adenoviruses most commonly cause respiratory illness. However, depending on the infecting serotype, they may also cause various other illnesses such as gastroenteritis, conjunctivitis, cystitis, and rash. Symptoms of respiratory illness caused by adenovirus infection range from the common cold syndrome to pneumonia, croup, and bronchitis. Young infants, and especially patients with compromised immune systems, are more susceptible to severe complications of adenovirus infection. Acute respiratory disease, or ARD, which was first recognized among military recruits during World War II, can be caused by adenovirus infections. Vaccines were developed for adenovirus serotypes 4 and 7, but were available only for preventing ARD among military recruits. However, vaccine production was eventually stopped in 1999. Strict attention to good infection control practices, including contact and droplet precautions, is effective for stopping nosocomial outbreaks of adenovirus-associated disease. Maintaining adequate levels of chlorination is necessary for preventing swimming pool associated outbreaks of adenovirus conjunctivitis, and frequent hand washing is recommended in group child care settings. There are several benefits of using adenoviral vectors for transferring viral antigens into host cells to trigger desired immune responses. For example, because of the relatively large-sized and well-characterized genome, adenoviruses are easy to manipulate genetically. Also, they cause mild infections in humans. Another major advantage of adenovirus-based vaccines is their ability to induce strong and sustained innate and adaptive immune responses. To date, several adenovirus vaccines are in clinical and preclinical trials. There are vaccines developed against HIV, Ebola, influenza, Mycobacterium tuberculosis, and Plasmodium falciparum, currently under human clinical trials. There's at least one adenovirus vector-based vaccine being developed for COVID-19. Adenovirus-based vaccines are prepared by inserting a transgene cassette into the adenoviral backbone through direct cloning or homologous recombination. The transgene cassette expresses the target antigen under the control of a strong promoter that is capable of maintaining robust and sustained expression of the transgene. So let's look at this process a little more. The most widely used method in adenoviral vector development involves homologous recombination between a shuttle plasmid carrying the transgene cassette and the adenovirus backbone. This strategy is relatively straightforward and can be easily performed with a variety of commercially available protocols, reagents, and kits. Generation of adenoviral vectors is commonly carried out in mammalian cell lines. A shuttle plasmid is constructed to express the 5' end of the adenoviral genome, in which the E1 transcription unit is replaced with the transgene cassette. The shuttle plasmid is transfected into a mammalian system alongside either a rescue plasmid DNA containing an E1 deleted adenoviral genome, which is designed to slightly overlap with the 3' region of the shuttle plasmid, or the wild-type adenoviral genome with cleavage in the E1 region. Removal or cleavage of E1 greatly reduces the risk of generating replication-competent viruses. Within the mammalian system, homologous recombination between the shuttle plasmid and the adenoviral vector leads to incorporation of the transgene cassette into the arboviral backbone. The shuttle plasmid can also be generated in such a way that the E3 transcription unit is replaced with a given transgene cassette. Replication-competent or oncolytic vectors are employed for cancer gene therapy. Oncolytic vectors are engineered to replicate preferentially in cancer cells and to destroy cancer cells through the natural process of lytic virus replication. Many clinical trials indicate that replication-defective and replication-competent adenovirus vectors are safe and have therapeutic activity. Here's an example of one of these oncolytic viruses. So remember, adenoviruses encode E1A and E1B that inactivate the tumor suppressor genes RB and P53. Loss of RB leads to cellular replication, whereas loss of P53 prevents the cellular apoptotic response to viral infection and aberrant cellular replication. Cellular lysis results from unimpeded viral replication. The modified adenovirus lacks the E1B gene and therefore can only lyse cancer cells in which the cellular P53 gene has been mutated. The presence of intact P53 in surrounding normal cells leads to their apoptosis following viral infection, preventing widespread infection of normal tissue.